Hi everyone. In today's video, I'm going to be going through the processes of glycogenesis and glycogenolysis. So firstly, before I go through them, what is glycogenesis? So glycogenesis is the main process that occurs in liver and skeletal muscle in the fed state. So essentially, it's the process of glucose molecules that have been absorbed from food being absorbed into the liver, skeletal muscle for use around the body and also in cells in skeletal muscle and other cells in the body through GLUT4 transporters, and in the liver and brain through GLUT2 transporters. And this glucose being transported and being converted to glycogen. So say, for example, we have a cell here, and this cell has its own plasma membrane, which is a lipid bilayer. So this plasma membrane, say if the cell is in the skeletal muscle, will have a GLUT4 transporter on its membrane. This GLUT4 transporter will allow for the transfer of glucose into the cell, and eventually it will allow for this, the conversion of glucose into glycogen. And this process has many steps within it, and I'll just like to go through the main steps so that you get the big picture idea of what glycogenesis is. So, glycogenesis. It's the genesis, the making of glycogen. So obviously, we have to start with glucose. So, glucose will enter the cell, as mentioned before, through GLUT2 or GLUT4 transporters. And first of all, it will be phosphorylated. So essentially that means that a phosphate group will be added to the glucose, and this will form glucose 6-phosphate. Now glucose 6-phosphate is essentially a glucose molecule with a phosphate group on the 6th carbon atom of that molecule. And you may be wondering, why does this actually occur? Well, essentially, because there's a concept of a concentration gradient in which molecules and particles always move down from a place of high concentration to low concentration, we want to maintain glucose flowing into the cell. That's why we phosphorylate it. This phosphorylation allows for a concentration gradient to be maintained into the cell, because essentially, as the glucose molecules enter the cell, they are disappearing, in quotations, because we're just converting them to a different molecule. And this process actually occurs because of two separate enzymes. In the liver, this occurs due to glucokinase. And in other cells in the body, other than in the liver, it occurs due to hexokinase. Now, after we've added this phosphate group to the glucose, this is not actually the proper form or the functional form that will allow us to form glycogen eventually in the cell. So we need to convert this molecule to something else. So, next, it's converted through a mutase enzyme to glucose 1-phosphate. And now you may be wondering, why does this occur? So glucose 1-phosphate is the molecule that is preferred in the formation of glycogen, as when the glucose molecule has this particular structure, it's able to actually polymerize and therefore form the long and branched glycogen chains. And the reaction that actually occurs is not like a phosphorylation reaction as we see here, but is instead called an isomerization. Essentially, mutase is able to move the phosphate group from one position, which is the 6th position, to the 1st position, or the 1st carbon. After that takes place, we have the formation of a special molecule that's called UDP glucose, or uridine diphosphate glucose. And this molecule is very important because not only does it allow for the carrying of this glucose 1-phosphate molecule, but it allows also for the glycogen structure to essentially form. And the enzyme that allows this to occur is called pyrophosphorylase. So just to recap so far, glucose, we firstly have glucose traveling into the cell, being phosphorylated, being converted to glucose 6-phosphate. That allows us to have a concentration gradient to be maintained into the cell. Then glucose 6-phosphate is going to be isomerized into glucose 1-phosphate by the mutase enzyme. Then it's going to be converted to uridine diphosphate glucose, which is sort of like a carrier molecule that allows for glucose 1-phosphate to easily be polymerized into the glycogen molecule. And this process is assisted by the pyrophosphorylase enzyme. So that's kind of like the second last stage in the process of glycogenesis. The next stage that actually occurs is just simply the formation of the glycogen chain. And this process occurs with the enzyme called glycogen synthase. And now, as we saw before, actually, this glucose molecule had a phosphate group on it. So if you picked up that glycogen does not have any phosphate groups, you'd be correct, because essentially glycogen is just many glucose molecules joined together. So how is this phosphate group lost? Essentially what happens is that this glycogen synthase molecule, in order to activate it, it must be phosphorylated itself. If you've ever learned about DNA or DNA transcription, you would know that RNA polymerase has to be phosphorylated in order to be activated. And it's the same concept here. Essentially, glycogen synthase, if you think about it, is able to take this phosphate group for itself, become phosphorylated, and therefore activated. And glycogen synthase essentially allows for these glucose 1-phosphate molecules, or which will become glucose molecules, to join to one another in order to form glycogen. So essentially, this is the overview or very basic viewpoint of glycogenesis. Moving on to the process of glycogenolysis, it's essentially the opposite of what occurs here. But I'd like to draw it separately because there are some stages that are actually missing. So, glycogenolysis, what is it? Lysis, as we all know from words such as hydrolysis, mean cutting apart a certain molecule. And glycogen is the molecule that we start off in this process. So, it's essentially just cutting glycogen. So, glycogenolysis is a process that occurs in the fasted state, as glycogen is actually the main molecule that's used in the body as a source of energy. When the body's not consuming any food, the GI tract is empty, and therefore it must provide energy for key molecules and key organs, such as organs of the nervous system, such as the brain, and also erythrocytes or red blood cells. So this glycogenolysis process first starts off with glycogen, which is obvious. So we have glycogen here, which may be present in the liver or skeletal muscle. And this glycogen is essentially cut up into small molecules, but this time the enzyme that does this process is called glycogen phosphorylase. This is different to the molecule that joins the glucose molecules together called glycogen synthase. So I mentioned earlier that glycogen synthase has to be phosphorylated in order to be activated. However, this time the result of glycogen being broken down is actually glucose 1-phosphate. So, essentially, because the product of the reaction of glycogenolysis is glucose 1-phosphate, and we have a phosphate group being added instead of removed in glycogenesis, this enzyme actually has to be dephosphorylated in order to be activated. Now, it's okay if this doesn't make sense for the first time. Practice is the key, but you just have to remember that because there's a phosphate molecule already present here, we essentially just want to take it off and donate it to this glycogen synthase molecule or enzyme. However, in this case, because we want to donate a phosphate group from or add a phosphate group to the glucose 1-phosphate group, in order to make a glucose 1-phosphate, this enzyme needs to be dephosphorylated in order to be activated. So if you think about it this way, it does make a little bit of sense. However, you can watch through this video multiple times in order for it to make more sense. So firstly, we have glycogen being converted to glucose 1-phosphate with glycogen phosphorylase. However, glucose 1-phosphate, as we even saw in glycogen synthesis, is not the functional unit of glucose within a cell. Now, the mutase enzyme that I mentioned earlier comes into play once again. So mutase, instead of converting glucose 6-phosphate into glucose 1-phosphate, now converts or isomerizes glucose 1-phosphate into glucose 6-phosphate. Now, something very important happens at this stage. The skeletal muscle in your body doesn't actually have the enzyme that will allow for the conversion of glucose 6-phosphate into glucose. So, in skeletal muscle, glucose 6-phosphate stays as is and can be used directly in the process of glycolysis in order for the production of energy. So essentially, this means that skeletal muscle has its own private store of glucose because essentially glucose has to be converted to, glucose 6-phosphate has to be converted to glucose in order to be sent around the body. If it stays as glucose 6-phosphate, it can only be used by skeletal muscle as it can't come out of that skeletal muscle. However, if we're in the liver, for example, we can convert glucose 6-phosphate into glucose. And this is done because the liver has the enzyme glucose 6-phosphatase. And a rule of thumb is that any enzyme that ends with tase takes away a phosphate group. So this essentially means that the phosphate group from the 6th glucose molecule, carbon atom, is removed in order to just form glucose. So in summary, the process of glycogenolysis is a lot simpler than glycogenesis because there's one less step. First, we dephosphorylate glycogen phosphorylase in order to activate it and therefore convert glycogen into glucose 1-phosphate. Mutase is able to isomerize glucose 1-phosphate into glucose 6-phosphate. And finally, in the liver, if we have glucose 6-phosphatase, we're able to convert glucose 6-phosphate into glucose in order to be used around the body or even in the liver for the process of glycolysis. However, if we're in skeletal muscle, because this enzyme is missing, we are unable to convert glucose 6-phosphate to glucose in order to be used around the body, and therefore the glucose 6-phosphate can be used directly as a source for the process of glycolysis. So in a nutshell, these are the processes of glycogenesis and glycogenolysis. Glycogenesis occurs in the fed state, and glycogenolysis occurs in the fasted state.